Alright, so the next section we're going to do is cognitive neuroscience. So how do we study all the things that we talked about in the last sections? Neurons and lobes and the hemispheres. Okay. So studying the actual brain. So cognitive being the thinking part, neuroscience being the pieces, building blocks of the brain. Okay. So where and how does thinking actually occur in the brain? So we've talked about the mind-body connection and how we should do models that match the brain. And so now we're going to get into how do we measure that. And so why is this important? And this tends to be a little difficult for people, so make sure you're getting this part. Converging evidence is that I have results from over here and results from over here, and they show the same conclusion. And so having replication, looking at several different ways to think about the same thing and make sure you get the same picture. So we wouldn't have labeled Broca's area if we didn't see that multiple times from several different things. So lots of psychology in general, not just cognition, is trying to make converging evidence where I have the two pieces or more that say the same thing. There's sort of a couple ways to do this. Dissociation is the ability to say that doing one thing affects one task but not another. When we get to transfer appropriate processing in the learning notes, the memory, how do we remember things section, we'll really look at how I can do one task that only affects one thing at once and not these other things that seem to be related. So we're dissociating them. We're saying that these two things are separate. Okay. A double dissociation, I don't know what happened to the font here, but it says that it's an activity that it affects one process and then has the reverse given a second activity. So I have these two different things that I'm doing. One time it affects it this way, another time it does the flop of it. And that's pretty important in the brain in the sense that if I have people performing a language task, I'll see a dissociation, so it affects the language part of the brain but not another section of the brain. And then we'll have them do a very similar language task thinking these things are the same thing. We'll get the same area of the brain and it'll flip. And we'll get something over here in the spatial area and we'll be confused. What just happened? So double dissociations help us understand down to the minutia of what is actually going on in the brain. But those two don't seem to be converging evidence. That seems to be just, divergent evidence. And that's actually useful for converging evidence because it lets us see what occur when associations actually happen. So the effects of doing something are related to doing this other task. So these two tasks measure the same thing, that would be converging evidence. These two tasks do different things, that would be diverging evidence. So both are pretty important. Alright, so the types of research. We're going to talk about three, and I would go back to that brain worksheet that I was using before. So there's behavioral research, that's the general come to the lab, do the stuff type of research. There's correlational brain research, where we're just looking at what goes on in the brain when we make people do something. Remember, correlational research is when I do have them look at something and then just measure, I'm not manipulating anything. And then there's causal brain research, where we're actually going to manipulate something, and usually it's because of a stroke or a lesion, an area of the brain that's broken, and not because we hacked out half their brain, okay, because people don't like that. So let's start with behavioral methods. So behavioral methods measure, it's hence behavioral from the first chapter, where we're measuring actual output on the system. So we can look at reaction times, we can look at whether or not people are getting it right. Essentially all of coursework is a behavioral measure. Can you prove to me that you know this enough to make an A, B, or C? So I have ways that I'm measuring these things. So we're going to use that data as either converging or diverging evidence from the brain data. So this seems like it's in the wrong chapter because it's really about how do we measure the brain, but we also need to understand the output of the system at the same time. So that's where that converging, diverging thing comes in. Having the output from the person, where they're guessing answers or watching a movie, whatever, and then looking at what's going on in the brain. So we're going to measure some observable behavior. And then two important terms. One problem you have with experiments is sometimes things are too easy and everyone gets it all right. And that's not very helpful because we want people to have a variation so we can see what makes some people better, what makes them worse. So if we make everything too easy, it's called a ceiling effect because everyone's scores are at the top. If we make something too hard, it's called a floor effect because everyone's scores are at the bottom. So I've got to find some type of task where I can get people in the middle. Okay. So for instance, I had a graduate student just telling me they were having people remember words across all these different conditions, and all he had was floor effects. People were getting 6 or 7 words correct. People should really get like 20 to 30 words correct, and he couldn't figure out what's going on. So he made his tasks too hard and thus was sort of stuck because all he had were people getting the same number of things correct. So that's not very helpful. All right, this crazy table is a little too much sometimes. So what I like to do is flip over here and have you make your own. Okay. So I've got this in the bottom of the new brain sheet, and I want to look at each one of these separately. So I don't really have slides for each one separately. I have just take some notes. Okay. So what is accuracy? Accuracy is just how much you're getting right. Okay. That can be percent, that can be number of items, so it's just a measure of how well you're doing in the task. So the good thing about accuracy is that it's objective. As long as you have a good way to score this that makes sense, you know what right and wrong are in this scoring system, it's very objective. So it helps us keep it straight what their scores actually are. Okay. The problems with this system, sorry, flip back and forth here, make sure I was on the right slide, is the ceiling and floor effects. So we can miss something because we've made the task too easy or much too hard. So the trick with accuracy is finding that right spot in the middle. Reaction times are how fast someone does something. Often called the speed of processing. So I want to see how quickly people can respond to a letter on the screen, or I want to look at how fast they can make some sort of judgment. It's all about the speed of processing. The good thing about these is they're also objective. Oop, I can't spell. There we go. So they're very objective. As long as you've got a good system to measure them, they can tell you about how fast things are. The problem with this is that you can't find the specific process. So what do I mean? If I have people responding to words as either yes, it's a word, or no, it's not a word, I'm trying to figure out how fast is lexical access. So here's my dictionary, yes word, no, not a word. But I also have the fact that I'm having them type their response. So they're entering yes or no, or they can say it aloud, yes or no. So I'm measuring both how fast it takes them to read the word and how long it takes their motor cortex to form the response to the word or to type the thing. So sometimes the processes are kind of tied together and you can't do the one process at a time. And then the other problem with them, I like to call the spaced out problem. When you're doing participant data, sometimes they don't want to be there, often a lot of the time. And so they'll just hit the 7 on the keyboard the whole time, so you have responses that are too quick. They couldn't have possibly read the screen that fast. I think you have people who like to stare at the ceiling, twiddle their thumbs, and so the other issue being that sometimes their reaction times are very long, 5-10 seconds. That's forever in cognitive years. Your action potentials are so fast that you don't know what to do. Like their responses are too short and too long, so you've got to find a way to deal with that issue. Alright, the next one is judgments. This is any of those teacher evaluations, the any type of scale, that stuff that's called a Likert scale. Most people say Likert. Like any of that 1 to 7 stuff, where I ask people to tell me what they think. And so one of the first problems with them is they tend to be very subjective. But they're a good way to measure those sorts of things. So it's a way to make introspection slightly objective. So I can get an average score across lots of people. So it is an introspection task, that's why it's slightly subjective. But it is a way to sort of put a number to those tasks. So again, they're slightly subjective. And then you can have sort of bad questions. So if you've ever read a scale and said, what the heck are they asking me? It's a bad question. So I've got to figure out a way to make sure that everyone answers it in the same way. So they all understand the question the same. And then sometimes the scales themselves are bad too. So looking at those teacher evaluations, this class was in the top 20% of all classes I taught, or I took rather. I teach statistics along with this class. Most people don't like statistics. I hear this a lot. So it's hard for people to say this is a top class that I took when they went into the class thinking, oh, why do I gotta take this? So sometimes you just have bad scales. Alright, protocol collection is sort of an odd one, used more in human factors or in industrial organizational research. It's a way to ask people about a task. So, can you explain to me how you got dressed and ready for this morning? Or I guess since we're online this semester, can you explain to me how you got your computer out and, you know, your coffee made to watch this lecture? So it's about getting like a series of events or a sort of a way that someone did something. Okay, so that automatically makes it pretty subjective, and it's hard to score. So you have to find new ways to sort of score these things. They're not hard and fast numbers. But the good thing about them is that they can reveal information you hadn't thought of. So if I'm asking someone how they do a task and they say something unusual, that is a new way to think about it. So I can ask how they perform this task and look at the sort of things that we hadn't thought that they were doing. So this happens a lot in like consumer research when people are testing new systems. They say, you know, if you had just moved the keyboard down an inch, it would be easier to use. Those sorts of things. Okay, so these are all the behavioral measures, and I have my good list and my bad list. You can also look at this slide that gives you an example. It's tiny, you'll have to blow it up. But it gives you an example of all of them. It says kind of what their advantages are, but also sort of their disadvantages. All right, so what are some other problems with behavioral research? And this is global. This affects every single one of those different types of tasks. Expectancy effects, or sometimes called social desirability, this idea that you know what's going on in the study, or you think you do, and so you're going to act differently than your normal day-to-day life. We have a problem with that in every single lab where people know they're in an experiment, so they act differently. It's kind of hard to get around. Task demands are when you're asking people to do something, especially if it's boring, to pay attention. So people get bored in the middle of it, they're tired of being there, you've asked them 100 questions, so asking people especially to do something difficult can make the later tasks you ask them to do different than if you had just asked them to do the later one. So you've got to kind of deal with the fact that the order of events matters and people getting bored matters, that sort of thing. And then the bad part about behavioral is, what is the brain doing? We haven't gotten to how the brain is working when we measure these. But remember, the important part about behavioral research is that we can use it to converge or diverge evidence for the brain research. All right, let's see. What's next? Correlational methods. All right. Correlational designs, remember from the research section, are when I look at two things at once and just say these things are related because they go up together, they go down together, or something of that effect. We're not actually manipulating anything. So pretty much all this next section of brain research is correlational because I'm looking at, I made them do this, here's what their brain did. But I didn't manipulate any of their brain, so I can't say that this causes this. But I can say this area of the brain looks like it's doing this. Okay, so that's what this correlation does not imply causation section is about. We're not saying that these two things cause each other, but probably they're related because we get this same effect across all these experiments and all this behavioral research. So we have this crazy chart, and it's going to have 4 different things on it. So spatial resolution, temporal resolution, how invasive it is, and then the cost. And I've got some definitions over here. So looking at what kind of correlational method should I use? So spatial resolution is how close it can get to where it is in the brain that's producing that signal. So we'll get to EEG work or electricity measures, and they're not very good at localizing. They say, you know, it's over here. That's a big section of the brain. So spatial resolution is where the brain is doing that action. Temporal resolution is how closely we can track changes over time. So you want a really small temporal resolution, like in milliseconds. So quick time actions, because most of cognitive processing is very fast. So we want to be able to measure that very distinctly over time. Invasiveness is how much it sort of hurts. Sometimes I call this the pain section for the person. So some of the older systems required you to be injected with radioactive dye. And then sometimes they're just annoying because they're loud if you've had an MRI. So how invasive is it to the participant? And then cost. How much does it cost? And I'm not really sure what the bottom of the slide is supposed to say, but the first 3 are super important for neuroimaging and then cost for each participant. So here's the mad crazy slide. What we're going to do is track all of this information over here on my little chart. Alright, so what have I got going on here? I've got my correlational measure. I have the abbreviation for them. You don't have to know the full names because they're really difficult to spell. So electroencephalogram, event-related potential, electromyography, that should be a G. Sorry about that. And then what it is. So the electric ones all tend to go together. So it'll help if you remember that they're electric. So let's start with the electric ones. And then I'm just going to pull up these pictures here. So an electroencephalogram, or an EEG, let's make this just a touch bigger for you. Is a system that measures the electricity from your brain. So remember, I really stressed that action potentials were electric, and that's super useful because now we can measure the electricity that's coming off your brain to look at what action potentials are firing and therefore see what the brain is doing. The aroused brain, the awake brain, alert brain, is got very fast little waves. It looks like a quick squiggly diagram. A relaxed brain waves slow down some. When you're asleep, you get much slower brain waves because you are not processing as much. You get these little K-spindle things. That is the sections where you're dreaming. And then in deep sleep, where you're like really out, you get very big brain waves. So we can measure how fast the brain is working. So I'm going to go back over here. So for time, for these electric ones, so all 3 of these, it is great. A+. So the cool thing about electric ones is that the time measurement on all of those is very quick. So I can measure to the millisecond or faster. let's go down to the next slide, an event-related potential. And I have a picture of an EEG cap here for you so you can see what someone in a study might have on their head. So this is the way I would measure those sorts of things. And then each one of these little white things is an electrode. They touch your head. They don't go in your brain. And it measures the electricity coming off of your head, which is supposedly from the brain, and all these cords are connected to a computer. So an ERP, or an event-related potential, is usually a second of data for an EEG. So what happens is people will get showed a picture, cute fluffy kitten, blown up building, and people's reactions to those are measured. So you'll get differences in the milliseconds after you see the picture depending on what you've seen. You can do this for language as well, and what happens is the specific waveforms change. So an ERP is a just very narrow focus on an EEG. It's like 1 second of the data. So let me go back here. So that's this one, an ERP. So let's talk about space. What do they do for space? They're terrible. So the bad thing about EEGs and any of these electric measures is that they are not good at spatial resolution. I can't tell if I can tell it's coming from like here, but not really the specific set of neurons. Okay, so they're pretty bad at this. Let's see, I have a picture for an EMG too, I believe. Oh, nope, just a slide. So electromyography is when you get wow, brain fart, the electrodes on your face, and it measures all the muscle groups of all the eye movements. You can also get them done on like your arm to look at muscles, several other places, but mostly it's done on the face, so you can study smiling and frowning and all these other eye movements. You usually get one here on the eyebrows, one here on where the eyes crinkle when you smile, and then some down here for the different smiling muscles. So it looks at the electric potential for muscle groups, usually on the face. So how much do they hurt? They don't really. It might give you a slight headache because usually those caps are a little tight, and they put some goo in them, so if you don't like goo in your hair, it's pretty gross, but generally they're not very painful. So that's good for the participant because you don't want them to be hurting. Okay. And how much do they cost? They're not very expensive. Expensive. Okay, now, they are several $1,000s. There are cheaper ones, but in the grand scheme of things that we're going to talk about for brain work, several $1,000s is not very much money. Okay, so really, this is the only thing that's bad, is that they are not good at spatial resolution. Okay, they are good at everything else. So lots of people like to use them. Come on now. There we go. All right. Let's move on to the blood systems. Is it going to flip? Come on, new slide. Okay, or not. There we go. All right. So all of the blood systems use the oxygen in the blood to measure where the activity is going on. And so the idea is that as blood moves through the brain, the oxygen needs to go to areas that need more brain functioning. So as I am thinking about something and that area of the brain is like, more oxygen, we're thinking really hard, more blood will move to the area, which gives us a better picture of what brain area is activated at the time. Sometimes it's called lighting up because these pictures often look like weather maps. Okay, so this is a really old school system. It's called a PET scan. And those were the ones where you had to be injected with a radioactive dye, and then they would measure the isotope decay. So they're pretty old. I mean, look at this computer. Oh my goodness. This is an fMRI. So it's an MRI machine, if you've had one of those done. You don't have to be injected with dye anymore, but they are loud. They are so loud. And they tend to be very constricting. They're kind of small. They have these new open air ones that are much better, but it tends to freak people out because they're so small and they're so loud. And they are magnetic, so people with pacemakers or other sort of, like if you have a plate, you can't use it on that area because they're magnetic. Alright, it's a little more. PET scan stands for positron emission tomography. Whew, can't talk. So this is that radioactive isotope thing. So any activated area has increased blood flow. And look at these little pictures. They're great. So as I'm looking here, they've got what they had people doing. So this will map onto what we did in the earlier section. So when I have people viewing something and hearing something, here's my temporal lobes in this red area, and the occipital lobe in the back. And then you get a little bit of frontal cortex because those things are loud, or you're sitting there thinking. If I have people close their ears, so they plug them up, you see less activation from this top left picture to this top right picture where the temporal lobes are. So that's kind of neat. If you have people close their eyes, you see from the top left picture to the bottom left picture, you see a decrease in visual activation in the occipital lobe, and you see an even smaller area if your eyes and ears are closed. Why do I still have this area at the bottom right where my eyes, I still get this occipital lobe activation? Well, even when your eyes are closed, so for example, close them, you still kind of see like purple hazy shapes. You're still getting visual input, it's just not the normal visual input. So that's a PET scan. An MRI looks more at the magnetic pieces of oxygen ions. And so generally those are used to look at, areas of the brain or the spinal cord or legs or whatever. So they're not just used for brain research. The ones used for brain research are functional MRIs. The slides are acting crazy. There we go. Let's flip back here. 1 second. There we go. So functional MRIs are more when you map the brain over a specific period of time. And so you get these pretty pictures, like the one on the bottom left, would be temporal lobe activation, where I've got my brain and I can make a weather map, so red is more activation and yellow is less, of what the brain is doing. So remember we talked about the 10% rule. And I said I would come back to why I think that comes in. These pictures look like you're only using a very small part of your brain. And that's a lie. So what happens is your whole brain is activated. You're thinking, you're like, oh, this is so loud. Why is it so tight in here? What do they want me to do? I'm seeing things, I'm hearing things. So the whole brain is on. And what they do is they take a picture of you just sitting there doing nothing, and then whatever they want you to do, and they subtract them out. And so what happens is you get these pictures like on the screen of just the areas that got the extra bump, or extra less, if you will, from you sitting there doing nothing. So these are pictures of the difference between you hanging out and you doing something. So that's probably one of the many reasons where this 10% myth came from, because it's how the pictures look. This one on the bottom right is going to have some of Broca's and Wernicke's activations, temporal lobe, and a little bit of that occipital lobe. Okay. The way an fMRI works is that it has multiple MRI scans, so it's lots of scans together. Alright, before we go on to optical imaging, let's go back and label our chart here. So I've got a PET scan, an MRI, and then MEG, which I haven't explained yet. 1 second. So MEG is a new system that is like an MRI++. So it's a faster, cleaner system for an MRI. So for time, our PET scans and our MRI scans are actually pretty bad. So they don't measure very quickly. because the scans each take 20-30 seconds or more, and so the timing on them is not so good. Meg is a little better, so we'll say it's in the middle. Keep my happy faces going here. For space, all three of these are great. So the whole point of a blood system is the space. So I can tell where something is occurring in the brain versus when it's occurring. Painful-wise, Meg is alright. So it's in the middle because it's still a system that is loud and kind of constricting. You have to lay there for a while, that kind of thing. The PET scan systems tend to make people very uncomfortable. And MRIs are somewhere in the middle. They're tolerable, but they still kind of suck. When it comes to money, though, Meg is like super duper expensive. So let's think about this in dollar signs of restaurants. It's a $4 restaurant. It's very expensive. PET scan is more of your $2.50 restaurant. It's expensive, but not too bad. And MRIs are pretty expensive. It's your $3 restaurant. They're pretty expensive. So you have to have a lot of money usually to do these types of designs. Alright, the last one we're going to get to is called optical imaging or optical tomography. So diffuse optical tomography is like the sonar for your brain. It's a laser system, and the light is reflected at different rates through your brain and the blood in your brain, and so you can track thinking. And it's super cool and very new. And so this idea of it measuring sort of like sonar. So optical imaging is great at the time functions. It measures pretty quick. It is great at the location functions because it's like sonar. It is not painful. Did you just sit in there? Although its downside is that it's very expensive because it's new. Alright, so those are the types of correlational measures. Let me go back here. Right, so we've done this one. Alright, what are some of the problems with correlation? All we're doing is measuring more brain action. Okay, so there's more electricity or more blood flow, but it doesn't really tell me if it's a positive or negative action. So it doesn't get it at that go, no-go system, and it does not really get at the idea of blood flow to an area does not necessarily mean like more activation. It just means you need more blood. So we're using that, but it's sort of a con of the system. So more doesn't equal more necessarily. And everybody's slightly different. So I've talked a little bit about how us lefties are weird. In the sense that Broca's is sometimes on the other side, or the brain areas are flipped. And so it makes it difficult to study because if you're looking for one specific area, it's cheaper if you just look at that one area. But if you don't know which side it's on because they're left-handed, it makes it more trouble for you. And everybody's slightly different. I pick on lefties because I am one, and I've had some neuro people say, well, you lefties are weird. So everybody is a little different. That gives you some trouble too. And then lastly, the brain is always thinking. There's no 10% rule. So you could be thinking about Swiss cheese the entire time the experiment is going on, and so they're not getting an accurate picture of what you're doing. That's why the behavioral measures help. It's a way to check and make sure people are doing what we think they're doing. But you never really know. Last topic, the causal brain. So we're going to talk about lesion studies, transcranial magnetic stimulation, and drugs. But again, this chart, not so great. So I'm going to flip back to my Word document here. I'll talk about all three of these. I have them in slightly different order, sorry. So, lesion studies. The very famous case for lesion studies is the Phineas Gage case, where, and I have that on a slide here, let's go on, here we go, where this construction foreman got a steel rod through the brain. it narrowly missed all the spots that would have killed you, and went through one of the cheek bones. And so it went through the frontal lobe, and basically people said he was a totally different person. I can imagine that would do that to you. But it was one of the ways to talk about how the frontal lobe is where most people think personality sort of exists. He definitely lost his job. He was quick to anger. He couldn't work with others, and he actually became a sideshow exhibit. They recently have released some pictures of what they think he looked like, and they couldn't clearly take the rod out, so they cut it right close to the face. But that's a lesion study. Most lesion work, not that slide yet, is done on people with specific, like a stroke. So if you have a stroke, that will give you a specific lesion, an aphasia, or a cancer where they've removed a tumor, those sorts of things. So the good thing about this is it gives us a very specific place of the brain problem. Not Brian, brain. So it gives us a very, I know exactly where the issue is. The bad thing is while that is specific, it sometimes crosses boundaries. So if I have an aphasia in Broca's area, it's often not that just Broca's is broken, if you will. So strokes, for some reason, really love Broca's and Wernicke's. But it often is Broca's plus some of the more of the front. So it doesn't always just devote itself to a very small space. It tends to cross spaces that we understand what the functions are. And then you do have to find the people to be in your study. So these are natural events that we use people for. They are causal because something has been manipulated in the brain, but we tend to have to just sort of find people. And thankfully, there's not too many of them, but then you have the issues of everything else that matters in your life. Diet and, environment, where you grew up, what kind of education you've had. So there's lots of other variables that can cause you trouble. All right, let's do drugs. So one's called Curie. I'm sure I'm mispronouncing that, but it's a drug that will basically turn off part of your brain. This research is done in animals because most people don't like having their brain turned off. And so the good thing about it is that it is non-permanent. And it does get at a fairly specific area. Oh, my spelling skills. There we go. Of the brain. And so I can sort of look at what happens when we turn off specific spots. It is very disconcerting for the research that was done on humans. It tends to freak people out, which isn't very helpful if you're trying to study them. For transcranial magnetic stimulation, if you've ever heard that you shouldn't hold a credit card next to a magnet because it'll demagnetize your card and screw it up, or it will, if you hold a magnet next to a computer, it screws it up. Your brain works the same way. So our brains are electric, and if you hold a big magnet next to them, you can screw them up. So again, it's non-permanent. It only scrambles your brain for a little bit, and we can get at a very specific area, but it is really upsetting for people. So the same basic problem. Maybe. There it goes. So those are all the causal brain areas. So why is that causal? Because I have manipulated something. Having a computer moment here. 1 second. We're going to look at this. Okay. And then there's some more information on the slide about how transcranial magnetic stimulation only works for about 1 inch, and some of the drug problems can be more than one specific system, and then time-wise. So it just kind of depends. This gives you some more information. Let me get to where I'm at here. Last but not least, models. So we've walked through, here's the behavioral work, here's the brain work. Let's finish with this idea of how can we map that into a computer simulation. So going back to the information processing approach. So what computer simulation models do is they take all that behavioral and brain data and make them converge. So we look at how to build a model that does what people do. So it's a little separated from artificial intelligence, although they tend to be tied together. Because artificial intelligence, the idea is to train a system to act like a human, whereas computer simulation models are more, can we get this to mimic human behavior so that I can test other little things? Very similar, like I said. Process models tend to sometimes be called box models. Because instead of being, here's how this brain system works, it's this system leads into this system. So it's more mapping those processing systems with their algorithms versus looking at the little pieces. So often called box models. Specifically for computer simulation models, we're going to spend a lot of time this semester on neural net models. Sometimes these are called parallel distributed processing models or connectionism. They all mean the same thing. So neural nets are models that are they often are called bubble models. You'll see why in a second. Where I have lots of interconnected units that's supposed to sort of match neurons. It's not quite the idea of a neuron, but it's supposed to be sort of similar to a neuron. So you have some sort of input layer. I am going to tell the computer that I saw barks and tails and fur. And that is the stimulation. So that's supposed to be like you seeing this thing. The input layer is connected to a hidden layer. And that's the part of the brain that we don't understand. There's lots of stuff going on in the brain at once. And so this sort of mimics the fact that we don't really totally know what the brain is doing. And then that hidden layer is connected to an output layer. And the output layer goes, oh look, dog, because of that specific input. So here's a picture. Maybe, there we go. So we have this input layer where I would see, barks and tail wags and slobber. The hidden layer would do all that processing. Here's all those things getting put together. And the output layer would say, oh look, dog. So this is a very simple neural net, but that's sort of the way a lot of the language processing systems work. An example, although not totally great, of a process model or box model is an example of the process that's going on. So I see some sort of probe. So in this study, they've got these numbers that you're having to remember. So I have to remember 1, 5, and 2, or the 7, 6, 2, 1, 8 thing. And then at the end, they're going to ask me a probe. They're going to say, did you see the number 4? And so I have encoded the number 4 in this second one. And then when I see the 4 on the screen, I have to scan and figure out if I saw it before, make some sort of decision, and then answer yes or no. And so that is a process model trying to explain this picture. So that's scan and compare as I get more symbols in memory that I have to remember. So 1, 3 here or 6 here. I'm going to take longer to answer in this decision section because the scan and compare spot has got more things to scan. So it's trying to explain this data, but not neurally, more just like conceptually. So that was a lot of information about the brain. So what do we got to know? Definitely understand the difference between the types of brain information: behavioral research, correlational research, causal research, and modeling. Like, understand what the purpose of each of those is. Be able to give an example of the types. So an electrical type would be an EEG, a blood type would be a PET scan or an MRI, causal research would be a lesion study, modeling research would be process or neural nets. So I give it some examples. And really use that table. So the tables that we created are supposed to help you organize all of that into like one little piece. So those tables from the book or the tables from the notes that we just took will really can help you take all of that information and sort of collapse it together.